Good afternoon, everyone. Today, actually, that was the old title, although it has changed to lipid nanoparticle delivery of dicer substrate siRNAs. I think it's much easier to understand than oligonucleotide. And here's the outline for my today's presentation. So I'm going to start with our solid lipid nanoparticle technology, then followed by our in vivo screening process. Then I'm going to spend a little more time on the characterization and optimization of lipid nanoparticles. Last but not least, the tumor delivery results with our optimized lipid nanoparticles. And actually, thanks to Dr. Walter Ruzinski in the morning, who has covered the challenges in siRNA delivery in great details this morning. So I'm just going to go through this slide very quickly. We know that the therapeutic application of RNA interference is very attractive, however, extremely difficult due to the presence of the following challenges. And to overcome these challenges at Dicerna, we have developed this solid lipid nanoparticle technology. As you see that this lipid nanoparticle is completely different from the conventional liposomes because this doesn't have the aqueous core engulfed by lipid bilayers. And these are the representative lipid components for this lipid nanoparticle, which can be further divided into the core lipid components and envelope lipid components. The core functions as an RNA magnets which interact with negatively charged RNAs, and the envelope lipids actually are critical for the transfection of targeting cells. And this is a schematic of our manufacturing process. As you can see that upon mixing, the pre-extruded cores will interact with negatively charged siRNAs to form this loaded cores. And then the envelope lipids will wrap up this loaded cores, followed by standard buffer exchange and the concentration process before being filled into sterile containers. As you can also see that this process actually is different from most other processes in the biotechnology field because there is no inline mixing, so you don't have to play around with Reynolds number. And this entire process is charged by this process is mediated by charge-charge interactions and active encapsulation of negatively charged oligonucleotides, which really allows the binding of high levels of oligonucleotide, and also the particles can protect the RNA in circulation and helps release of payload into the cytosol. Although it's much quicker to get in vitro results, but due to the discrepancy between in vitro and in vivo results, so we screen most of the lipid nanoparticles on animal models directly. This actually is a summary slide that we prepared in 2012. Over here, you can see that by fine-tuning the compositions of lipid nanoparticles, we can make those particles relatively inert in the liver or highly active inside the liver with IC50 around 20 microgram per kg. And this formulation, although it's quite inert in the liver, it's actually a very active tumor-centric formulation. The other interesting observation is, as you see, the company name Dicerna is based on our technology, dicer substrate siRNA. By simply converting the 21-mer siRNA to dicer substrate, which referred over here as 27-mer, the potency in liver was improved by almost threefold. And we all know that upon 12 injection, all the particles will go to liver first before being distributed all over the body. And over here, what you can see is by fine-tuning the composition, we can make the particles relatively inert in the liver, however, very active inside orthotopic hep3b tumors. At 6-hour time point, there is not much knockdown at all in the liver, but there is already significant knockdown on hep3b orthotopic tumors. Our first clinical program is in HCC patients, liver cancer patients, which will be launched second quarter this year. And this slide just shows our capability of delivering oligonucleotides to multiple types of HCC models on animals. Among them, hep3b, hepG2 were implanted as cell suspensions, and HH1, HH7, and MHCC97H were implanted as tissue fragments, which are much more difficult to deliver. And at that point, we have sort of fully optimized the lipid particle compositions. The question we ask ourselves is, not changing the particle composition, can we still make the delivery even better? That's the reason we started screening of manufacturing processes. And as you see that, this actually is the process we use to manufacture our prototype LNP. And this LNP was used to produce all these results above. Compared to many other processes, this is still pretty good until we identified this one. Upon single IV administration at 5 mg per kg, 80% of the target knockdown was achieved in Hep3B orthotopic tumors. But when we compare particles manufactured by the two different processes, the particle size, polydispersity, and RNA encapsulation actually were quite similar. But anyway, since we have observed this improvement in target knockdown, we carry out efficacy study. This part of the slide was generated with the prototype LNP. And just some background information that all these tumors were sorted based on AFP level before dosing, because these are orthotopic tumors, so they were implanted into the liver, not sub-Q models. And the final tumor weight range actually from 1.5 grams to 3 grams. Those are really huge tumors. And with prototype LNP, we can see the ED50 value was about 5 mg per kg. However, when we test the particles manufactured using the new process, the ED50 was improved to about 0.5 mg per kg, which is almost a tenfold improvement in potency. What we also know is over here that most of the time, the clinical candidate or the clinical lead may not be the most active compound you have, but rather the one which gave the broadest therapeutic window. So we decided to carry out tolerability studies to prepare formulations manufactured by these two different processes. You can see that 3 batches of materials were tested at very high concentrations, 10 mg per kg dose, BIW 2 weeks, so total 4 doses. The results were kind of shocking because with prototype LNP, some severe toxicities actually were observed with animals, severe body weight loss and severe liver weight elevation. But these two particles, those are the particles manufactured by the new process. This one is manufactured in-house, and this one actually is manufactured at our contract GMP manufacturer. Those two formulations were very well tolerated. We have also evaluated those blood markers which are associated with liver toxicities, because all these parameters actually are used in clinics to monitor liver toxicities on patients. As you see that, again, the prototype LNP generated some severe toxicities on those animals, but this was not observed with lipid nanoparticles manufactured using the new process. Now keep this in mind, that these two formulations, actually, they have the same composition. They have same amount of lipids, same amount of oligo, similar particle size, polydispersity. Basically, everything at this point we have tested cannot differentiate those two particles. And then what is the real difference? Which brings the next section of my talk, characterization and optimization of lipid nanoparticles. So these days, dynamic light scattering has been widely used to measure particle size, and this is just typical size distribution using DLS technique. As you see that there is just not much difference between these two types of particles. What we also know is, since this is a size distribution, so there will be some subpopulations underneath this fitting curve, which should look like this. The question would be, is there any difference within these subpopulations? That's the reason we carry out the work with size exclusion chromatography to separate these populations based on their size, collect all the fractions, and then measure the particle size again. Now we can see that with the prototype LNP, a small peak was observed with fraction number 5, but this was not observed with the new lead LNP. And these days, most particle size was measured under intensity mode because people believe that this is more representative to the hydrodynamic diameter of the nanoparticles. But what we also know is, under intensity mode, large particles will weigh a lot more than smaller particles, simply because the mathematics, the formula they use to calculate particle size, as well as the properties of those large particles, they can reflect a lot more light. That's the reason we decided to collect even finer fractions, and then measure the particle size under intensity mode, volume mode, as well as number mode. This is the size measurement, size distribution of the parental particle under intensity mode. And now we can see that after we collect those finer fractions, some smaller peaks were observed with the prototype LNP associated with those later fractions. However, in the volume and number distribution mode, we found actually those smaller particles are the majority in this formulation. And here's the particle size distribution measured under number mode. You can see starting from fraction 4, all these particles have much smaller particle sizes, which no longer like liposomes, but more on the micelle range. But this was not observed again with the new LNP manufactured using the new process. Now we have two questions. Since this is just formulation, and then where is the API oligonucleotide with these fractions, and what are the lipid distributions? Volume distribution these days are believed to be more representative to the mass distribution. And that's the reason over here, particle size was displayed under volume mode. And you can see that with the prototype LNP, only 36% of the siRNA was associated with fraction 2 and 3, which gave particle size between 79 to 106 nanometers. But the majority of siRNA actually was associated with later fractions in the micelle range. With this improved process, we can see that this process actually yields much more uniform particle distribution. And all the RNA payloads is associated with this LNP fractions in the lab, with the size in the liposome range 51 to 78 nanometer. Now, another question is, since we observe this separation between the micelle peak and LNP peak or liposome peak, the question is, does the micelle fraction also deliver? So we first evaluated the liver knockdown of LNP and micelle fractions. We can see that both fractions really deliver the oligonucleotide to the liver. However, our target is liver cancer. So we have also generated results based on tumor delivery, although I cannot show it over here. But to achieve efficient liver tumor delivery, you would really like to have more these LNP fractions. But this is also in the context of our LNP composition. Now, this is a new schematic of our formulation process, which actually is the same as the old one. What we have really optimized are just these numbers: the order of addition, ionic content strength, lipid and RNA concentration, water content in each every step, which is critical, and dilution factors. As I said, another intrinsic limitation for DLS technology is, regardless of particle shape, it only gives you a diameter, because the assumption is everything has spherical shape. So we did this analysis using cryo-TEM. And as you see that the pre-extruded cores do have the conventional liposome structure, which has the aqueous core engulfed by lipid bilayers. However, although the polydispersed is small with the final nanoparticles, still 3 populations were identified over here. Particles with this complete solid structure, and solid particles engulfed by this lipid bilayer, which have this bullseye structure, and particles don't belong to either of those two categories. We also did analysis on the particle size distribution, circularity, and populations based on over 3,000 nanoparticles. These particle size distribution results actually correlate very well with number distribution. And most of particles, as you see, do have spherical shapes. 75% of particles have this type 1 structure, completely solid lipid nanoparticles, and 13% have this bullseye structure, and the rest of them do have this micellar sizes. All right, in a biotechnology company, you can probably just keep characterizing your particle, but still at the end of the day, you need to show some efficacy results to the board. And we generated great results previously using Hep3B orthotopic model. That was at the end of 2012. The first experiment we did in 2013 is to see if this efficacy was reproducible. And again, you can see that the ED50 on Hep3B orthotopic tumors is about 0.5 mg per kg. And the rule of thumb for most of anti-cancer drug is as long as safety allows, hit the target as hard, as frequently as possible. However, what we also know is RNAi is a long-lasting effect. So is 3 times per week really necessary? And what we found is actually 1 mg per kg was equally efficacious when given twice or 3 times per week. And also, again, particles manufactured in-house or at our GMP manufacturer showed similar efficacy results. Alpha-fetoprotein has been widely used in clinics to monitor the tumor treatment progress. And we can see the final AFP level actually correlates over here very well with our anti-tumor efficacy result. At this stage, 2 questions we need to answer. So to optimize the best dosing regimen, where the dose saturates or where the knockdown saturates and how long the knockdown really lasts. So we did these experiments. And from here, this is a single IV administration. We can see that the knockdown saturates between 3 and 5 mg per kg after single IV bolus administration. And at 5 mg per kg dose, the knockdown in orthotopic Hep3B tumors lasts over 7 days. So next study, we combined all these results and did this dose optimization study. And what's impressive over here is at 3 mg per kg, once per week administration is efficacious enough. But to achieve the best anti-tumor efficacy, it seems 3 mg per kg, twice per week administration is preferred. And again, this anti-tumor efficacy was confirmed to be on-target knockdown of MYC. MYC was used as a therapeutic payload over here. Not only we can inhibit the growth of tumors when implanted in the liver, also our formulations can successfully deliver the oligonucleotide to Hep3B tumors when implanted subcutaneously. And the β-catenin over here was used as a therapeutic payload. This is actually a very popular payload used in treating liver cancers these years. With all these results, I would like to conclude the following, but to save more time for questions, I'd just like to point out this. DLS need to be used very rigorously, and cryo-TM is very useful, however, very expensive, time-consuming, and low throughput. And all these results just demonstrate the potential of optimized LNP RNAi therapeutics for targeting traditional undruggable targets in oncology. And Dicerna is currently a public company now. We just launched our IPO in January. But still, we consider ourselves as a small biotech, so we'd like to acknowledge everybody in the company. And thank you. If time allows, I would like to take any of your questions. Do we still have time?
We have exactly 1 minute. I'm sorry.
Yeah.
That's a good presentation.
Thanks.
My question is regarding the charge on the particle liposomes. Are these anionic or cationic liposomes?
If you paid attention to the structure of those lipids we use in the formulation, all these are ionizable lipids, which means at low pH they are positively charged. At physiological pH they will be neutral to have the optimized circulating time and minimize interactions with blood component.
So basically, like these are in low pH, and when they enter human body,
the initial buffer in the process is acidic. Eventually, a TFF tangential flow filtration will actually exchange this with a neutral buffer. So at that pH around 7.4, those formulations will be stored and dosed to patients. And at circulation, during the circulating time, those particles will remain electroneutral. Only after they enter the endosome, they will become positively charged.
Okay. Yeah, thank you.
Sure.